Well, it's that time of the year, folks. Not leaf peeping season or pumpkin spice season or even apple picking season. It's respiratory virus season. The symphony of sniffles and coughs is just around the corner. And it's that time of the year that we in the clinical lab are stocking up all of our supplies for respiratory virus testing, figuring out what the heck we're going to do from an algorithm perspective, and putting communications out to try to convince people that every kid with the sniffles does not, in fact, warrant a highly multiplexed respiratory virus test panel. So the diagnostic landscape for respiratory viruses has evolved dramatically in the last 5 years with widespread institution of molecular tests in labs of all sizes, but also at the point of care, and even consumer direct testing. So what's next for respiratory virus testing? Where can we go from here? Welcome to Editors in Conversation. Please subscribe to this podcast and share with your friends and colleagues. This episode is brought to you by the Journal of Clinical Microbiology, available at asm.org/jcm. Editors in Conversation is supported by the American Society for Microbiology, which publishes JCM. If you are a member of ASM, you can get up to 50% off the publication fees when you publish in JCM or any of the ASM journals. I'm JCM Editor-in-Chief Romney Humphries, and I'm joined as always by my co-host, Dr. Elli Theel. Hey, Elli.
Hello. So, Romney, before we
Yeah.
get into anything, I have never in my life heard of leaf peeping. Is
Are
that
you
like
serious?
is that like you just go look at the
Yeah.
leaves, right?
Yeah.
Okay.
Okay, so I only heard about this last year, but it was all over the news. And yeah, you go look at the leaves. Yeah, it sounds a little, I don't know, illicit,
Sounds
going looking at
wrong.
leaves.
Leaf
Yes.
peeping.
Leaf
Anyway.
peeping. That's the thing. You'll have to make sure you do it.
Hmm.
Yeah.
Hmm.
So, Ellie, I have a question for you. Let's be
Yeah.
honest. When your family has the sniffles, do you test them?
Not since 2021. No, I don't. I just assume it's something that will go away in a couple of days. So we don't routinely test. What about you?
Oh yeah. Every time we test for quality control.
Oh. Hmm. So that's where we differ.
It is where we differ. Even more exciting is when I get to test myself.
Mm.
Anyhow, so today to talk about respiratory viruses, we're joined by Catherine Hogan and Ben Pinsky. Catherine is an assistant professor in pathology and lab medicine at the University of British Columbia and the BC CDC Public Health Lab. Catherine, welcome to Editors in Conversation.
Thank you for having me. This is good natural timing as I just am recovering from an upper respiratory tract infection myself. And I'm happy that this is occurring today and not yesterday. I'm not sure I would have made it. So thank you for having me.
And okay, did you test yourself?
Yeah.
What it was?
I was not able, I don't think I would have been able to make it to the lab, and I haven't tested myself yet. I have not also profiled any of my nasopharynx for metabolites, which we can talk about.
Okay, we can talk about that. And we're also joined by Ben. Ben is editor-in-chief of another clinical microbiology journal that we won't mention, the Journal of Clinical Virology. We'll try hard not to hold that against you, Ben. And Ben is also a professor of pathology, infectious diseases, and pediatric infectious diseases at Stanford, where he's the medical director of the Clinical Virology Lab. Ben, welcome.
Thank you. Thank you very much for having me.
Wow, pleased to have you guys. So, Ben, maybe we'll start with you. You and I, we started our careers at about the same time, right when viral cultures were going away and more molecular testing was happening. And in fact, fun fact, the only reason I got my very first job, which was at UCLA, was because Ben, who had also applied for it, turned it down. So, for people who... Thank you, Ben, for turning down the UCLA job.
I think it worked out well for both of us.
I do. I think so too. Yes, I was the sort of consolation prize, I guess you could say.
No, not at all.
So molecular testing's dramatically changed how we do things in the lab. I mean, I no more drum rollers and viral cultures and all that exciting stuff. But there are some challenges with molecular testing. And so maybe you could just kind of tell our audience what some of the limitations that you've seen in your practice with molecular testing for respiratory viruses are.
Sure, yeah. I mean, definitely molecular diagnostics for respiratory viral infections have sort of revolutionized testing for these analytes. But there are some challenges. I think the main one that I think we deal with is there are so many different options, many different panels, panel sizes, speed of testing, and trying to figure out what the right test is in the correct clinical context and sort of optimize that, I think is a big challenge for our current diagnostics for respiratory viruses.
Yeah.
I'm not sure, though, that our discussion about metabolomics is going to resolve those questions.
Narrow the option.
We'll add yet another option potentially to the arsenal of diagnostic testing.
Very cool. So Catherine, follow up for you as well. Do you see the same challenges in your setting? Where do you think the real limitations are?
I agree with Ben. I mean, we're definitely seeing similar challenges. I think I would definitely highlight that for the immunocompromised host population, just as a subset, I think it's been particularly challenging. We do routinely see individuals who have persistent detection, through nucleic acid testing, and I would just highlight that as one of the examples where we would, or at least we wish that we had additional diagnostic tools to help inform on clinical relevance, amongst other scenarios.
Yeah, absolutely.
Hmm.
So the premise of the study that we're going to talk about that you all published in JCM was that infection with a specific respiratory virus results in a distinctive metabolomic profile that can be harnessed for diagnostics, maybe even prognostics, and help bridge some of the gaps that we talked about, especially the one you just mentioned, Catherine, right, where we're just able to detect viral RNA forever. So maybe you could walk through what exactly that means. I was going to pretend like I understood this, but honestly, once I got to like the multiple different types of mass spectrometry, I kind of lost the plot a little. So yeah, do you want to let us know what exactly do you mean by a metabolome, and how does that change during a viral infection?
Yeah, thank you, Romney. I appreciate your candor and just the opportunity to go back to the basics on this topic. And I would just say upfront, of course, Ben and I are here, on this discussion, but just through the manuscript that we put together, this is very multidisciplinary approach exactly for that reason. So if we bring it back to metabolomics, so I think you just asked, well, what does metabolomics mean?
Mm-hmm.
So in a nutshell, metabolomics is the study of small molecules that we call metabolites. And for humans, you can think of these metabolites as basically footprints of everything that's happening inside your body. So using specific instruments in the laboratory, which we'll refer to as mass spectrometry instruments, we can both measure and analyze the levels of these metabolites in different samples. So for metabolomics, we'll first perform untargeted testing or profiling, where we'll try to look at all detectable small molecules or metabolites. And from those, we'll try to identify what are some of the key features or key metabolites of interest so that we can transition to even simpler instruments. And I think what might be helpful is an example, just closer perhaps to current microbiology practice, where you can think of certainly some similarities with metagenomics, right? Where you can do metagenomic next generation sequencing, you cast a wide net in a discovery effort to glean some insights that can then inform the development and design of targeted testing. And I would just add, I think one of the reasons metabolomics is so compelling is because it's one of the disciplines that's considered closest to phenotype, in that it may provide the clearest picture of a current condition. And it really provides a layer of information that's not otherwise available through the study of genes or proteins. And that's also what makes it inherently more challenging.
Thanks, Catherine. That was helpful. I think it's I like how you compared it to metagenomic NGS. I think we can all relate to that. So can you tell us a little bit about what was known going into your study around metabolomics in patients with COVID-19?
Is that one for me?
Ben, I'm sorry,
Yeah.
Ben.
Yeah, absolutely. This project actually came up due to a conversation that I had with our uh, uh, staff scientist from the biochemical genetics lab, which is right across the hallway from our virology section. And I said, wouldn't it be interesting if we just put some NP swab viral transport media on your mass specs? And he agreed with me after a while and said, well, let's see what's in there. And so that's kind of where it started. It was actually before COVID-19, and we were looking at influenza, which I think we'll get to in the next question.
Mm-hmm.
But the data on metabolomics for SARS-CoV-2 infections, I would say there's a reasonable amount of literature. It's quite mixed. And most of the studies are actually on serum or plasma, looking at what metabolites are in those specimen types. And that's kind of, those are kind of more typical specimen types to look at metabolites in. It's what your biochemical geneticists are much more used to and what they're used to putting on their mass specs, and they're not worried about messing them up. So the data there, again, quite mixed. I think one of the things that came out of those studies was a reduction in tryptophan, one of our amino acids. And so that had been consistent across a number of studies. It's thought that might be due to altered immune signaling, reduced biosynthesis of tryptophan, and potentially increased metabolism, potentially by gut microbes. But all those connections, are a little bit, I would say, still under investigation.
Gotcha. It's kind of amazing, the projects that come about from these random water cooler type conversations. So that's pretty cool.
Yeah, absolutely.
How much convincing did it take to get them to run this on their mass spec? I'm just imagining this conversation, Ben.
To be honest, not much. Tony will try anything, and so as long as it didn't ruin his mass spec, I think he was okay with giving it a go. And then actually, something came out of it. So it's fun to have colleagues like that.
Yeah, absolutely. So kind of getting back to it then, you talked a little bit about metabolomics and COVID. So what was known about other respiratory viruses going into this study?
Yeah, so we had done an initial study looking at the metabolomic profile of influenza-infected individuals from their nasopharyngeal swabs, and had done a sort of a similar analysis with quite a bit fewer number of samples. But it did a similar study and found some metabolites that could differentiate influenza from other respiratory viruses. And the metabolite that we found there was pyroglutamic acid, which may be involved in glutathione metabolism and probably has something to do with reactive oxygen species.
Ben, I feel like you're going to try and make us all biochemists. I don't know if I'm ready for this.
I don't know. I have enough trouble pronouncing all the compounds, so don't worry about it. And we won't show any structures.
No structures.
On this
good,
podcast.
That's going to bring back some serious trauma for me. So
right. We're no organic chemistry.
Thank goodness. Oh man. So Catherine, with all that background, right? So, we think these changes to the metabolomic, I keep saying it that funny, but anyway, that metabolic profile from patients who have infections may differ a little bit, and that may be helpful. So maybe you could walk us through your study design. Like, how did you conceive this study, and kind of, what did you look at?
Yeah, thanks, of course. So we did, as Ben mentioned, we did have some insights from some earlier work into some general principles. So we followed a framework that is very much similar to what what I considered at the early steps, just for the general pieces, but then we brought in some specifics for the question at hand. So basically what we did is, this was at Stanford Healthcare, both the adult and the pediatric sides. And what we did is to select individuals who were known to be infected with a respiratory viral infection, with a focus on SARS-CoV-2, but with a plan to also investigate other main respiratory viruses of interest, namely influenza and RSV. And we tapped into the large collection that we had of residual upper respiratory tract samples, mostly nasopharyngeal swab samples. And then we went by, and this is where it comes back to the earlier concept, is we started with the profiling on a discovery cohort using untargeted mass spectrometry on these samples, and we identified several thousand features and compounds that were present in the samples. And on those data, we performed machine learning analysis to understand how could this information be used to help infer which individuals were infected versus not infected. And from the several thousand compounds, we zoomed in on the top 20 features of interest that supported that differentiation. And after that, we performed targeted testing. So we used only the features of interest that we then moved to simpler type of mass spec testing. And we did the same thing again. We basically reran testing on a different instrument and applied machine learning analysis again to look at what was the test performance of those models to differentiate those infections and non-infected samples. Now, it's quite challenging, but it's not enough to just say these are the features that we found, because these features are basically described by things that are commonly used in mass spec. So basically they're referred to by their molecular weight and by their retention time on the column. So what we did next is we basically worked with the chemistry insights from our team to identify what those compounds were. So we basically proceeded to match those compounds to available databases to identify putative candidate biomarkers. And just to bring it back to clinical microbiology, we also performed finally SARS-CoV-2 viral culture that we combined with testing by targeted metabolomics to help confirm the findings and to explore them in that different way.
Gotcha. Thanks, Catherine. So you touched on this. It's a really important thing, but hard to understand, I think, which is the AI modeling that you used in the study. And so I am a simple person. So at a really high level, can you explain exactly how it works? So you mentioned that AI was used to identify features that could be targeted in an assay. But is AI also used as part of the final assay, or is it just to identify those features?
Yeah, and it's a great question. So just to begin, I would just really like to highlight Afraz Khan, who is one of the co-authors on this paper, who's a bright data scientist and a machine learning expert at BCCDC, for his insight. And if I have the courage, I'll send him the link to this podcast and he can review me for accuracy on this tough question. But just in brief, so we use machine learning, as I mentioned, to analyze chemical data from the patient samples. So as I mentioned, is when we actually do mass spec testing in this way, we generate thousands and thousands of data points. And what we want to know is, from all of these data, which specific features of those are linked to the label of interest, in this case, respiratory viruses. So what we do is we train a machine learning model to look at these patterns, to learn from them, and to then infer, once we actually present the model's new samples, to see, well, can the model actually help predict who is infected versus not? And to do this, we use two sets of data. We start with a dedicated training set, and then we follow that with a dedicated testing set to basically assess accuracy. So what we did is we used 4 different types of models to see which features or which small molecules were most useful to identify infection. And then we used an additional tool to show, well, which markers influenced the model's decisions the most. So to get it back to your question, Ellie, the machine learning part here was really used to identify features that could be subsequently targeted in targeted testing, but it was also used to predict infection status and to assess test performance. So it basically was used for both purposes.
For both. Got it. Got it. Okay, that helps. So you mentioned you use 4 different AI models, and they had some fun acronyms, LGCM, Lasso, so on and so forth. So are these really different models? Like why 4, or why those particular 4?
Yeah, a good question. And again, I'm not sure I can do it full justice, but I'll give you my take on it.
General is fine.
So basically, there's 2 things. So I think high level, for someone like me whose this is not his main topic, but we work really closely with our colleagues. Basically, the high level approach is that we use 2 of the 4 models were traditional statistical models. And you might have some, statistics background. You've seen these across projects, etc. So that includes lasso and logistic regression. And these models are characterized by their making certain assumptions. And one of those assumptions is they would be assuming a straight line relationship, for example, between data and outcome. Now, because those models may be very well suited to certain data sets, but perhaps not as well suited for other more complex data sets. So that's why we used an additional 2 advanced machine learning methods. So random forest and light GBM, so light gradient boosting machine, is in this podcast helpful, hence the lovely acronym. But I think what's really interesting just for us to say, what is high yield for us in this discussions? And actually, I think what I at least take away is for these 2 machine learning models, they're very interesting because they're based on decision trees, and they vary slightly in how they work. But basically, you can think of these as enacting flowcharts that ask yes or no questions to help sort the data and parcel out the data, and more importantly, to glean patterns in the complex data to which the model is subjected. So the intent of the multiple models is actually to build in an assessment of how robust they are and how reproducible the findings are across different models, and also to test at the same time how well the models react to certain types of data set. What was interesting for us is that in the current work, very similarly to what we had seen in earlier respiratory viral work that Ben alluded to on influenza, we saw that the top performing model was LGBM, so one of the 2 advanced machine learning models. And I would just say that as an additional layer to all of this, if we're not already fully saturated in machine learning, is that we also drew, what I should mention is that from the main analysis, notice that this was being used for binary classification, right? So it was yes infected, not infected, but we also added an additional layer to support multi-class analysis, meaning if we wanted to go beyond SARS-CoV-2 alone, which we did, we also drew on additional models, namely neural networks and SVM, support vector machine modeling, to help us look at more than 2 groups at any given time.
Fascinating and complex.
Yes. So what I got from that is it's helpful to have like bioinformatist AI scientist friends.
Best friends.
Yes, best friends.
Yeah, it was very important to have a Froz on the team. He did a fantastic job.
Mm-hmm.
Yeah.
And I'm very glad that you gave those questions to Catherine and not to me.
As I said, I haven't yet run them by a Froz, but it's just to your point, I would just say it's fascinating how things that we, read through or take for granted or I'm saying are similar, you speak to your colleague, of course, very different approaches. So I agree.
Oftentimes I skim through that part in the paper.
Yeah. Well, I had to chuckle a bit when I was rereading it because you guys went to a lot of effort to explain all the mass spectrometry terms that you were using, and then you just like name dropped all these AI models. So it's oh man, I know how that feels though. It's just like, and then magic happens. Cool. So Ben, let's turn to you. What did you guys find with all this? Like, what were the compounds you identified as top ranking for SARS-CoV-2 infection, and were there things that you identified in people who didn't have COVID infections?
Yeah. So this, I think Catherine mentioned this in the study design part, but it's actually quite challenging to go from the retention time and mass overcharge that you get out of this sort of experiment to the actual compounds. So you go into all these different databases and try to match up your signal to what's in those databases. And the databases may not be as complete as you would like. And then in addition, really to prove that you have identified a particular compound, you need to have that compound purified and run it over your particular assay and make sure that you get the same results from your clinical samples. And so we did that for a small number of the top-ranking compounds that we identified. The first one was 3-oxo-icosanoic acid. Sorry.
Here, you thought I gave
There's a
you
long
the easy
fat-
question.
Yeah. I can never say it, but it's a long chain fatty acid. It has 21 carbons. And the oxo, is the I said I wasn't going to talk about chemical structures, but it's a double bonded oxygen. And so it's in the 3 position, and we were able to identify that with some help from this. We couldn't buy commercially, so we had to ask our colleagues in chemistry to synthesize it for us, and they were nice enough to do that.
Wow.
And so we were able to identify that this compound is elevations in this compound is associated with individuals or samples that are from patients with SARS-CoV-2 infection. And that was the strongest association. And the second compound that we identified was commercially available. It was a little bit easier and it's also a little easier to say 2,4-hydroxyphenylethanol, also known as tyrosol. And so that one actually is decreased in samples from individuals infected with SARS-CoV-2. So those were the 2 top differentiating compounds. In addition, actually, some of the intermediate compounds are probably different isomers of that long chain fatty acid. So it seems like that pathway is particularly important in the nasopharynx for COVID-2 infection.
And so does that marker make sense based on what we know regarding people's response to COVID infection, SARS-CoV-2 infection?
So we do know that fatty acid synthesis is sort of disrupted in SARS-CoV-2 infections. Exactly, what the role is of this particular compound is not entirely clear, and that will require additional work. And similarly for tyrosol, it has some weak antioxidant activity, but that sort of pathway, it's not entirely clear how it fits in with the, what sort of mechanistic thing is going on in SARS-CoV-2 infection.
Yeah.
Interesting. Question, are these metabolites persistent throughout the patient's acute disease, or do they change? Did you guys get a chance to look at that? Like if you tested a patient in day 2 or 3 post-symptom onset versus later on, would you expect there to be a change in the metabolomic profile? Or no? I don't know.
Is that one to me or to Kathy?
I guess I'm sorry. I'm not doing a good job of directing my questions, but
I would say that we should investigate that question. I don't think we necessarily have the data to support that, but that's a super interesting question about whether these are persistent, what the sort of time course is of these particular markers, and does it depend on the time after diagnosis and what stage of infection they're in, or whether we can use these or other markers to determine whether a patient and predict whether a patient will develop severe disease versus have a mild infection. And so those are certainly questions we could attempt to ask with this sort of method.
Cool. So, Ben, you took the time to validate the compounds you identified as being important using viral cell culture, which I had to chuckle when you guys said you discontinued viral culture like eons ago. We just discontinued it a couple of years ago. But anyway, why was that particular component of your study important to do?
Yeah, it's another layer of confidence that these are, these compounds are important, and are true compounds differentiating COVID-2 infection from not infected. And we worked with the INOV lab, and this is when, we still had to do BSL-3. So they did all of that work to give us the cell culture supernatants from infected SARS-CoV-2 infected cell lines.
Yeah.
And also part of the reason was I was actually quite surprised at how well this worked. We haven't really talked about that, but the area under the ROC is like 99 or 100. We're talking about sensitivities and specificities that are greater than 95% for this sort of approach, which initially I thought was like, this is something else. Like this is somewhat surprising. So it was helpful, at least for me, to believe the results more, that we could show this also in an in vitro system, that the level of this long chain fatty acid was actually quite elevated in the supernatant of infected cell lines.
Cool. Interesting. Yeah, so you kind of had to convince yourself that what you were seeing was real.
Right. Yeah, exactly.
I love it.
We had a large number of samples. I can't remember the exact number. Maybe Catherine does, but we can look it up. But yeah,
People could read your paper to get that.
yeah. Read the paper.
Read the paper. Read the paper.
So we thought with a large number of samples, we would eliminate any confounding factors or things that we weren't thinking of that might not be,
Right.
that might give us the good answer, or, give us an accurate answer, but might not be, biologically relevant. And so this was very helpful to
Right.
convince ourselves of that.
Makes sense. So, Catherine, did the markers you identified for COVID also predict influenza or RSV infection, or did you see differences in those patients? And I know you guys kind of touched on that again, but if you could dive deeper.
Yeah, it's a good question. And I think, just to emphasize that we're kind of balancing two sides, right? So we said that this is a discipline that's closest to phenotype. So we'd like to isolate for a given virus and try to keep other variables, in check. But at the same time, we recognize that real world scenario is that you're not going to see SARS-CoV-2 alone. We're interested in other respiratory viruses. We're interested in co-infection. We're interested in bacterial co-infection, et cetera. So we were really curious, about this aspect specifically. And I think I mentioned earlier, but we had the original discovery cohort was devised looking at SARS-CoV-2 positive versus negative samples. But we did go beyond that for the analysis, specifically just because of the interest that we had. So what was interesting is that when we did expand the analysis with that fancy machine learning you've heard about earlier, what we were surprised, and again, to Ben's point, we were just surprised, like, how is it that it's working well, and does that expand to others? And we were seeing a very similar pattern. So we were looking at AUCs of 0.97 for influenza A, and 0.99 for RSV. So we definitely saw that there is some potential there that is not to SARS-CoV-2 alone, but to other of the main respiratory viruses of interest. And I think it is still hypothesis generating in that the study was not originally devised to address that, in that the signature wasn't derived for that. But I think it's really interesting to see, and I think it does spark additional questions. I would be particularly interested in better understanding what is the specificity of the signatures. And we've been toying with that question over time. Are we picking up on a viral specific signature, viral versus bacterial versus other? Or do we have the capacity through this approach of even picking out, between viral differences that would be sufficient, through differences in the metabolites to identify? So I do think that this will require more work, but it's been very intriguing for us to see those findings.
Yeah, I mean, absolutely. The challenge always is we can't tell if someone's infected. All we can say is there's a virus there, right? And so like this really is exciting. So Ben, as you were doing the study, right, we've talked about how flu and RSV, does that make a difference? The predominant subtype that you guys tested was Omicron variants for SARS-CoV-2. Do you think that would make any difference with different variants, or do you think based on what you're seeing, it's probably pretty consistent.
Yeah,
Or maybe we don't know.
we've definitely seen consistent results amongst the various Omicron subvariants.
Mhm.
We would really need to go back and look at the earlier variants to see, whether there is a difference, say, in the early days with, alpha and so forth.
Yeah.
And I'm not sure, and there may also be differences, in the patient population at that time that might be revealed from that, right? It would have been folks almost certainly been their first exposure, whereas later on it may have been, a second exposure or after vaccination. So I don't
Right.
know that we know that for certain.
Yeah. So kind of along the lines of different responses, Catherine, we often think of kids and adults responding to viral infections maybe a little bit differently. Did you see any differences with respect to metabolomics relative to patient age?
Yeah, it's a very good question. And we also thought that was quite important to explore. It's interesting that you take the virology, naturally, insight into this, but I would just couple that to the metabolomics specific question, which is we recognize that there are actually important age-related differences, particularly amongst the very young. So yes, we were really interested in looking at that. So in our study, what we did is that we predefined age and sex as a priori defined potential confounders that we wanted to look at. And when we incorporated age as a standalone variable in the multivariable model, we basically saw that it was not significantly associated with SARS-CoV-2 infection status. But I wouldn't discount that altogether, just because I think, especially for the very young category, the study did not include a high proportion of very young infants and children. It did include about a quarter of individuals, that were less than 18, so pediatric, but it just did not really tackle the very early. So I think that would be something that would be helpful to look at in more detail in the future.
Sure.
Yeah.
And what about race? Did you look at that? Did that maybe make a difference?
Yeah, so unfortunately, we didn't have the data were just very incomplete for race, so we were not able to capture that. But we recognize that this would be very important to look at in the future. And I would just add to that I think part of the limitations of the current design is this is a retrospective study. And so I think what would be really, really just make a big difference for the future is to integrate this type of testing into prospective studies that would allow better data capture for a number of variables, including race and ethnicity, potential other variables, social related to social determinants of health, vaccinations, therapeutics, etc.
Yeah.
Makes sense.
Yeah. Yeah, fascinating. So with that, Ben, this was all really interesting. So where do you see this work going? Do we think this might be used? You kind of touched on maybe bacterial infections, or, are we all gonna go out and invest in LCMSMSs for our labs? I actually did just buy one of those, but for my toxicology lab.
Yeah, you have those in the lab, the
Yeah.
LTMS, MS. Yeah.
Mm-hmm.
Yeah, so when we first pitched this idea to attempt to get funding, Catherine and I pitched this as a potential for point of entry testing. Like, this was at the height of the COVID pandemic. And the idea would be similar to when you get pulled out of line at the TSA and they swab your hands and your phone and they stick that thing in what is a mass spec, but just not the kind of mass specs we're using here, to look for material that could be explosives. So the idea would be here that you could use a portable mass spectrometer, which do exist and can detect the compounds that we're identifying here, and have a swab and put that directly into that portable mass spec and have an answer in a minute or less. And I think that's one of the advantages of this, although, mass specs are super expensive. It's very complicated to keep them running. But the sample prep for this is just to make sure there's no particulate matter in it, and to add a solvent, at least if you're going to use HPLC. And you can avoid all of that if you just put the swab directly and ionize it directly from the swab. So that was the idea.
When you said portable mass spec, I just imagined a little mass spec in my hand.
Yeah, like isn't the column length important?
They're actually not that. They're like tabletop. They're not too small. They're not too big, but they're not. It just
Yeah.
Small microwave size, something like that.
Yeah. So Catherine, same question. What do you think the future would potentially be?
Yeah, thanks. I mean, this is my favorite question, right? And I think it'll vary based on how ambitious you feel on any given day and how realistic you feel on any given day, you know, to tackle this agenda. I mean, I'm an infectious diseases physician as well, so I would just say very candidly, and don't let Ben hear this, I trained in virology with Ben, is I don't think viruses are the highest yield application. I think they're super important to look at. But I mean, like, honestly, molecular does actually pretty well for viruses on the most part, right? What I would like to see this in is I would like to really use, you know, this pilot. I do still think that there are important uses, there's no question, and joking aside. But I would love to see this expanded to those areas that are literally Achilles heel of microbiology testing. Like what can we do to address TB diagnostics? What can we do to address major caveats in syphilis diagnostics and syphilis prognostication, in neurosyphilis? Like there's a lot there that I would love to see it expanded to. And we're working on doing some of that. So we started some projects specifically for syphilis in particular. Can we combine it with other modalities? This is for metabolites, but I'm really curious in seeing, could we also complement this with lipidomics? Very similar, but still different, with procedures that are optimized for lipid recovery. And I think there's a few other things, you know, that really come to mind as well. Just completely agree with what Ben has framed, you know, earlier as to potential other uses, looking at portable applications. I'm in BC now. One of my colleagues here, for example, is running like portable mass spec in his van to detect compounds in salmon, which is like amazing. So where I'd like to see this go is, and back to your earlier comment, like talk to some friends and just see who does what are the needs, and what can we do. But basically, there's a lot that's out there for potential adaptation with ambient ionization techniques. So things that are just make it a lot more suitable to nearer patient, close to even point of care. And as an extension of the point of care, you know, continuum that we envision is, could we even adapt this to dipstick type testing? Right? Very similar. Could we identify compounds of interest that could then, through input with chemistry, R&D from industry, et cetera, could we adapt it even to simpler pregnancy-like testing, you know, dipstick style, for example. So, I mean, there's so much to do, I think, in this area. The question is, what do we want to tackle? You know, how do we broaden interest in this field so that we can really meaningfully try to work on some of these pieces? But I do think that there's a lot of potential.
Well, I'm going to put in a plug that you guys focus on urinary tract infection diagnostics. If we get a dipstick for that, that would be amazing. Well,
Noted.
guys, I mean, this was absolutely fascinating. And all joking aside, I really appreciate you both breaking this down for our listeners. I really think that there's a lot of potential in this for the future. And so this has just been fabulous. So thank you so much for joining us.
Yeah, thank you for having us.
Thank you very much. We would love to offer you a poster of the Krebs cycle for your office in memory of this discussion.
Yeah.
Oh my God, are you kidding me? My kid.
Inspired by our colleagues.
Yeah. Our biochemical geneticist, Tina Cowan, has an amazing poster of all the metabolic pathways in the cell. So we'll get those out to you.
I would love that. My kid is in his first year of college doing that Krebs cycle, and he was very scathing that I could not remember all the steps.
Oh my goodness.
So yes, that's awesome. Well, thank you again, guys. So if you've enjoyed this, please subscribe to the podcast and share with your friends and colleagues. And thank you for listening to Editors in Conversation. If you're interested in the paper we discussed today, and I absolutely recommend that you read it, it's fascinating. The link is available in the show notes or at asm.org/EIC. Thank you and goodbye.